Thanks very much. Thanks so much to the organizers for inviting me. I've been really excited about this conference since the beginning, and I'm very excited to be able to present here. So today I'm going to talk about really one project which is ongoing with Rachel Lee, Ben Engelhard, and Alana Witten. And in particular, it's really about one, I think, rich data set and how we can come to terms with it in the context of a sort of long tradition of models that I've been involved with for a while. And that's these reward prediction error models of the dopamine system, right? So that's a long-standing, I think, success story in computational neuroscience, going back really to Reed Montague, Peter Dan, Wolfram Schultz in the 90s. And I think it's broadly viewed as one piece of the puzzle of how learning works in the brain, and of course it's oversimplified, and a lot of what we're going to struggle with today is exactly whether it's oversimplified too much and how so. But I think there's something to it, right? And so what I'd like to talk about today are really two issues with these models, which I didn't, until seeing this data set, think really had anything to do with each other. Hopefully I convince you that they do. One is maybe the biggest conceptual problem with these models, and something that there's been a lot of attention to from people like Sam Gershman, Yael Niv, is the question of state. So these models are about making reward predictions and conditional on the sort of situation, the sensory inputs from the world, choosing actions. And a huge aspect of that, of course, is how the brain represents the input to that, the state or the situation, the perceptual problem. And in some sense, this problem sort of buries all of perception, and so all the issues that Mate talked about this morning, hopefully we can sort of ask Mate to solve them, and we'll sort of wire it into the dopamine models and sort of solve the problem separately. But in the meantime, in these models, people have tended to use very sort of simplified kind of oh, it's not showing my, hang on one sec. It's showing very simplified representations of sort of stripped down representations of the state of the task that people have tended to use to sort of simulate these models, and I think it's going to turn out that's not good enough for the data. The second problem is that this idea of prediction error is sort of the powerful idea is that there is a prediction error. It's sort of a thing that's broadcast. It's useful sort of throughout the brain. Dopamine is this tiny little set of nuclei, and they have this sort of diffuse ascending projection. And it's becoming increasingly clear, and the same I'm going to talk about is just one example of this in the last few years, that the dopamine neurons don't all do the same thing. There isn't really one signal. In fact, there's really extensive evidence of heterogeneity of variation between areas, between target areas, between sort of subnuclei of the dopamine system, and even from sort of neuron to neuron, as we'll show, within a single nucleus. And this really challenges, I think, a core idea of these whole models, and the question is what we can do about that. And what I'm going to say today is that at least some of what we're seeing in this sort of second problem actually reflects the first problem, that is how the brain represents a sort of complex, high-dimensional state sensory input to the system. And once you realize that, I think actually gives us a new way of a window of seeing from the perspective of the prediction error system what it knows about the state and what the brain is giving it. And so I think it's a new insight, hopefully, into these problems. Okay, so that's the background. By a further way of background, I want to start by trying to argue that this idea that dopamine is a sort of single thing, that the prediction error is a scalar signal that you want to broadcast, isn't just a straw man. I mean, at some level, of course, there's not a single signal, and variation isn't surprising, but it's also a deep idea that there's a lot of reasons that people were excited to take it seriously, at least sort of to a first approximation. And there's sort of both conceptual ones and empirical ones. So the conceptual ones are the whole notion of decisions, right, is really about boiling down all of the attributes of your choices onto a sort of number line so you can choose which thing to do. So the classic idea of Bernard's ass, he's got to choose between grain and water. How can you even compare those two things? You've got to sort of reduce them to a common currency. There are these theorems, beautiful theorems in economics that say that basically if you make choices that satisfy just basic conditions of rationality, that's equivalent to ordering the prizes on a number line. So this relates to stuff that Jeff presented yesterday, actually, about relating policies and rewards. So this is a core idea, and the way it comes up in reinforcement learning models is that you have an objective function, right? You have rewards, like utilities, every moment or every outcome is linked to a sort of scalar number, which is how good this is, and you want to optimize something, the so-called value function, right, which is this expected sum of future rewards, and you want to choose the actions that maximize that. So you sort of reduce everything to a common currency, and then you choose the action with the highest value, right? And again, it's sort of necessarily then a sort of scalar quantity. The individual rewards are scalar. The expected cumulative future value is scalar. And the prediction error for learning to predict future value from trial and error is also scalar. It's the difference between the reward you expected and the reward you got, more or less. The one thing that isn't scalar, right, is the state, right? So you start with a potentially high-dimensional kind of complicated sensory input from the world. That might be a sort of vector or feature, some kind of population code, but you're going to reduce that to a prediction about future value. You want to sort of project it down to this scalar thing. And so that's sort of core in how these models work. And empirically also, it was thought that dopamine was a good candidate for this idea of a common currency for a couple reasons. One is that just anatomically, as I mentioned, it's got this really kind of diffuse characteristic where these individual neurons, and there are not very many of them, relatively speaking, have very diffuse ascending projections that arborize over big areas of the brain. And as early as 25 years ago, for instance, people like Carl were saying, for neural value systems to constrain movement, they should broadcast their responses to wide areas of the brain, release substances that can modulate changes in synaptic strength. So this is sort of the idea that you could imagine neuromodulators playing this role, like dopamine. And then when Schultz started recording from these neurons, he reported, although without a lot of quantification, that the responses were very homogeneous, right? So, 90%, 85% of them would respond to reward. 70% of them respond to cues that predict reward. He tended not, because of this, he tended to display very few examples of individual neurons and lots of average data. And so we didn't have a lot of quantification for a long time what the variation really is, but it was taken to be surprisingly homogeneous. Okay, and this led to a sort of cartoon model, which is the one I'm going to start with, and which is the one sort of that comes out of the Peter, Diane, and Reid Montague's work, that basically the prediction error signal, we can sort of model it with a single sort of population of dopamine neurons that all display this kind of prediction error response, right? And then that is kind of one synapse upstream from that. There are value neurons, right? And again, there's sort of one of them, or one population of them, that represents expected future value, and that is associated with this kind of ramping activity in striatum that you see, particularly ventral striatum, prior to rewards. And it also trains the inputs to those medium spine neurons and striatum, the dopamine signal. It affects plasticity there between sort of cortical or cortical hippocampal neurons and so on that project to striatum and represent, broadly speaking, the state of the world. And dopamine trains the mapping from those to value. So you have this sort of compression at the cortical striatal stage from everything down to a prediction of value, down to an error in value that trains that prediction of value in kind of a closed loop. So that's the original story, and obviously it's oversimplified, but it's sort of a useful cartoon. Okay, so here's the main experiment I'm going to talk about today. This was done in Alana Witten's lab, mainly by Ben Engelhard. And it's a sort of classic perceptual evidence accumulation task. So an animal, let's see if I can get the movie to run. Of course I can't. The animal, well, if you can see the movie, the animal, there it is. The animal runs up a sort of tunnel. He sees towers on his left and right, and his job is to turn to the side with more towers, in which case he'll get juice, right? And there's sort of a noisy mapping from where the juice is to the towers, and the animal's job is to count them. So this is a sort of spatial variant on the classic evidence accumulation tasks. But the main difference here is this was done in this fancy head-fixed VR arena, so the animal's actually running on a ball, and there's a big screen around him, and he's in a 2-photon microscope, and there's a lens over his VTA, so we can record or Ben can record lots of individual dopamine neurons at once and really see what the population is doing. It doesn't record a lot at the same time, but there's lots of animals, so in the end we have 303 neurons, which is a pretty good data set, and a reasonably complex task for this sort of thing. So behaviorally, the animals, of course, can do this task, mainly summarized by this psychometric curve. So the x-axis is the difference between the number of towers on the right and the left, and the y-axis is how much they go right. So the animals, of course, can sort of tend to go right more when there are more towers on the right, so they can accumulate evidence, right? So this is not surprising, but it works. Okay, so I pretty much got involved in this project when Ben Engelhard showed up in my office, where there are, of course, no mice anywhere, just computers, and he said, well, we did this experiment, how can we analyze the data? And I said, well, I have only computers. All that you need to know in this class of tasks, there's only one thing, that the mice are going to care about. It's the difference in the number of towers, right? The key to these tasks is just keeping track of one number, the one that's on the x-axis of this graph, right, which is the number of towers on the left minus the number of towers on the right, or maybe even the absolute value of that, right? And it turns out, because of this graph, right, and this has been seen in monkey dopamine neurons and stuff also, animals of reward expectancy basically just depends on how balanced the count of towers they've seen is as they run down the trail. So for instance, this is a relatively hard trial. The animal starts out expecting reward about, this is the value function, the animal starts out expecting reward about 75% of the time. And as he sees towers on the left, on the right, and the left and the right, this is the running difference in counts, he's actually sort of ramping down his expectancy because he's getting to a situation where the difference is only 2, and so he's going to get it wrong 65 or 75% of the time, as you can see in this chart, right? So this is a hard trial, and that means the animal expects less confidently to get reward. That gives rise to a prediction error, which is sort of positive when he gets sort of confirmatory evidence and negative when he gets disconfirmatory evidence. And again, it's kind of balanced here. There's lots of negative errors because the animal's getting sort of bad surprises that things aren't as clear as he thought. So that's sort of the key prediction in this kind of task. Here's an easy trial where almost always the difference in counts turns out to be 6, and so the animal just gets more and more confident that he's going to get it right because sort of hiking up the psychometric curve, right? And you get lots of positive prediction errors. Now, just one more complication. We tend to think about time discounted future reward, so we don't just plot the probability of getting reward at the end, we plot it discounted by the time till I get there, and so the value is always ramping up, but it's ramping up less high if the ultimate reward is going to be occurring with less probability. The prediction errors they reweight a bit, but they don't change very much in this case. So this is what we think dopamine neurons are going to do. This is the average over all hard and easy trials. Easy trials, they expect a lot of value, and so the prediction errors tend to be positive over the course of the track. And you see this nice separation. Okay, so the key to this is that everything comes down to just counting this one number, right? The difference in counts. And that really is what we think the dopamine neuron should care about and should drive the prediction errors. And then one more thing, at the end of the experiment, or the end of the trial, when the animal turns left or right, we see this sort of classic prediction error. You get response to reward versus non-reward, modulated by how surprising that reward is. So in particular, hard trials where the animal was less certain to get the reward, you're going to see a bigger response to reward relative to non-reward. Okay. So here I am in my office with no mice anywhere, and Ben says thanks, and he goes away, and this has nothing to do with what he saw in the data set. And he was like, thanks, Nathaniel. We did our thing. Here's what the neurons actually do. And here's what the neurons actually do. And the answer is sort of everything. And I was justly disappointed in this, but this is the state of affairs. So there is good news. So the good news is, at the time of the outcome, when the animal gets to the end of the track, there really is this classic reward prediction error response. So something like 85% of the neurons respond more to reward than non-reward, and those responses are further modulated by whether the trial preceding it was hard versus easy. So this is 5 clusters I'll explain in a second. I think 4 out of the 5 significantly, the reward response is modulated by test difficulty. So the model actually does a good job of predicting what happens when you squirt the juice or don't squirt the juice in the animal's mouth at the end. However, during the time when the animal's running down the track, the bad news is the neurons just respond to sort of everything, and there are lots of variation. So again, these patients, there's really just one kind of prediction error, and it basically just cares about proximity to reward and whether the trial's hard or easy. But what you actually see is that some neurons respond at different points in the track, some respond according to how fast the animal's running or other kinematic variables. Neurons respond to cues, but they tend to care about whether the cues are contralateral or ipsilateral to the animal, which doesn't really have value consequences. It's a weird way of kind of breaking it up. Other things as well. And so this is what Ben did is sort of painstakingly identify different behavioral variables that modulated the response of these neurons, did a sort of cluster analysis and assigned neurons to clusters based on which cue they responded to, and ended up with 5 clusters given by these things. Here's another way of viewing the data. So a bunch of neurons respond to kinematic variables, some respond to position, some respond to previous reward, and so on. You can see that they're pretty well, and this cross-validates and stuff, they're pretty well separated in terms of caring about one or maybe 2 of these things, but they're really separate populations. All of those populations care about reward, as I mentioned. They're also anatomically separate, so this is sort of location of these neurons, and there's sort of significant separation between the neurons that care about kinematics and those that care about position and so on. Okay, so this is just a complete mess, and I mean this to insult the mice and their brains and so on, and not Ben and Alana. It's a beautiful data set, but it's a catastrophe from my perspective in my office with no mice anywhere. So what's gone wrong here, or how can we think about this? These pesky mice. Okay. So here's our prediction error, right? This is the sort of classic equation that has been used to think about the responses of dopamine neurons. And it really has 2 parts, right? The responses to rewards are driven by the sort of the immediate reward signal. The prediction is the difference between the reward you got and the sort of expectancy-related term. And then the expectancy-related term has a sort of forward-thinking part to it, which drives responses to cues that predict reward later. And so this is what we think is going to be driving, on a prediction error count, whatever's happening as the animal's running up the front of the maze where he's not getting any reward. So this is 0, and this is presumably doing stuff, and sort of fluctuations in that, like the situation getting better or worse, are going to drive cue responses. And one thing we can see immediately, which is really interesting, is that there's lots of different models in which people have thought about the possibility of vector prediction errors. And one of those, or one class of models, basically the vector is inherited from, you have lots of different kinds of rewards, and so you have lots of different kinds of reward prediction errors, the outcome time that give rise to different kinds of values, and so you have a sort of vector of different classes of dopamine neurons that care about different versions of R. And so one version of this is that Sam has worked on recently, Gershman, successor representation, it's called, predicts that basically you have variation here that in the outcome responses that's inherited and turns into sort of variation in the value response. So that's not, I think, what we're seeing here in this data set. What we're seeing is the reward really is a scalar. The neurons all really just care about whether you've got juice or not, but there's all this variation in the value. And so that sort of has to be, I think, coming in through the sort of state part, the inputs on which value is predicted and not on how it's sort of bootstrapped back to predict reward. And so I think that's one thing we can learn immediately. What else went wrong? So one thing is, again, I'm sitting in my office. I'm just going to keep saying this, no mice anywhere. And how do I turn this off now so I can play the movie? At some level, you know, the animal running up the maze is just counting towers, and that's just a number. But if you think about it from the animal's perspective, it's actually pretty rich, right? And this is actually not running as fast as the animals run, but this is sort of what it looks like. And it's actually, and plus he's kind of scrambling on the ball because it's hard to balance. So I think the whole thing, it's a lot, there's a lot more going on than you might be tempted to think if you just had a MATLAB window open. And in particular, it's just obvious the animal's experience is richer than a couple of numbers. And we all know, right, that the brain cares about all these things, and there's going to be all kinds of activity all over the brain about all these, you know, squares on the wall and the motion, and just, you know, that's obviously going to be the case. And so some level we shouldn't be surprised that some of that makes it down to the dopamine neurons, right? So one issue is just that we've oversimplified things in taking the kind of ideal observer model of the state. The other thing is the sort of anatomical model underlying this story is also sort of obviously oversimplified, right? The idea that you take this sort of rich representation in cortex and you just sum it all up into a weighted sum into a scalar value, and then you difference that into prediction error, that's a sort of nice computational story, but we know that's not how striatum and dopamine look, right? It is actually the case. There really is a sort of convergence as you go from cortex to medium spine neurons and from medium spine neurons to dopamine and then back. But there's also a lot of topography, right? So the cortical striatal projection really does preserve kind of broad, the famous loops and so on, topography from one to the next, and then that topography, there's also topography from the striatum to the dopamine neurons and back again. And so at some level, it can't really be the case that these neurons all do the same thing and these neurons all do the same thing unless all of cortex does the same thing, which it obviously doesn't. And so that's, again, maybe just a little too simplified. I think all you have to do is sort of put those two things together to sort of solve the puzzle of this data set. In particular, if the connectivity from here to here to here to here is anything short of like perfectly uniform and convergent so that all the neurons get the same inputs, then any variation that's in the cortical code, as it were, broadly speaking, is going to be sort of reflected in variation in the striatal code and variation in the dopaminergic code, just like a funhouse mirror, right? It couldn't be otherwise. Just going to sort of inherit that population code, even if you don't change anything else about the model or how you interpret it, right? And so in that respect, basically the reward prediction error response in the end is going to show you a window kind of on the feature code that's 2 steps back from it in cortex, which is the state that we're kind of interested in, okay? So let me just unpack that a little more. So one thing to say is, sorry, this is the learning rule for the weights. They get updated according to the prediction error at each step. Imagine another extreme, and you could go anywhere between these two, where there's one striatal neuron for every cortical neuron, right? Turns out because the value prediction was assumed to be linear in the features, which is traditional in this kind of model, if you get value from features by adding them up, then you could just defer that sum one step because everything is linear. You could have neurons that specialize in the value consequences of one particular feature from the cortical code, whatever that is, and then you have a whole bunch of little pieces of value functions, and then you add those up at the next stage to get prediction error, right? And it's because you actually haven't changed the model at all, right? This model computes exactly the same thing. It's just sort of mapped onto the cartoon circuitry in a different way, right, taking advantage of linearity. We could repeat that trick, right? We could have one dopamine neuron for every value channel, and then we have a sort of mini sort of partial prediction error that computes the prediction error consequences of that feature relative to part of the reward, and then gives it back. And as long as you sum up the rewards in the ascending step and train all the weights that way, then again, we haven't changed anything about the model. Okay, so that's, interestingly, then we can take, this sort of classic 1997 algebraic model that we thought we'd rejected and just sort of map it differently, slightly differently onto this sort of circuitry, particularly kind of respecting this topography at each stage, and predict, a seemingly vector error signal that's really just carrying the scalar information. And of course, you don't have to have full separation, or you have partial convergence, or any sort of combination. The only thing that you really need to keep the model, the sort of classic model the way it was, is that at the end, you really do have to sum everything up by the time you get back to doing the plasticity. If you don't do that, everything sort of still works, but it makes different sort of behavioral predictions, and these, I hope, will be testable. The features represented in those different channels, so things like kinematics and position and cue responses and stuff, they won't block one another in the sort of classic Rescorla-Wagner sense of blocking. If they don't share a prediction error, they'll learn fine, but they won't compete to explain reward. And so that's, I think, something we can actually test to the extent that these channels remain separate. If they converge, then there's no problem. And I think it is probably the case that there is a lot of compression at the stage of sort of dopamine to striatum. At the last kind of ascending stage, the dopamine arborizations really are huge. There's like a single dopamine neuron here that's filled and imaged and is measured in millimeters. covers a lot of striatum, and that's just one of them. And there's a sort of extrasynaptic propagation of dopamine volumetrically, so that overall, to the recipients, I think it probably does kind of average over the neurons a fair amount. And again, it doesn't have to be perfect, but I think that's the thrust of the story is probably that there really is a lot of convergence on the way back up. Okay. And again, what this highlights is that this sort of input vector, whatever it is, this sort of feature code, the thing that we've been sort of pretending in previous models was sort of a labeled line for the state of a task or a count of numbers, is going to get carried through this whole circuit and is going to cause all kinds of variation across neurons. And I think that's basically what we're seeing. Okay, so that's the account. So let's talk a little bit about how it explains the particular data then. So the first thing is, whatever this feature vector phi is, it's going to drive, if it's different from neuron I to neuron J, this part of the value function or the partial value function, changes in that Q I multiplied by some weight are going to drive a prediction error response at Q time, even if the reward is 0. And so that means that whatever phi is, sort of arbitrary input sensory variables, are going to correlate with different neurons corresponding to those variables, either because W is actually, say, positive because phi is value relevant, you're ramping up as you get closer to the reward or something, or even if not, if it's completely irrelevant, just because W is sort of not going to ever be 0, it's going to fluctuate, and so you're going to see, with learning, you're going to see some responses to the cues, basically, that dopamine neuron cares about. So that thing is where the cue responses are coming from. And at the same time, it also explains, because the reward is just the reward across all the channels, why all of these channels, basically, whatever cues they care about, also all care about the reward. So basically, all the neurons, or most of them, will respond differentially to reward than non-reward. So this sort of weird split between the cue response and the reward response is explained by this. Okay, so to make this more concrete, I'm going to move to sort of a deep network model. And again, because I'm sitting in my office and I don't have any mice, for me, like, this task is basically the same as the mouse, monkey, whatever it is, task. Here's a task that we can train a deep network to play to sort of exemplify the structure of these computations and these predictions. So obviously, it's not quite the same task that the mice are doing, although close enough as far as I'm concerned. But it has this sort of property that it's sort of continuous, high-dimensional, and there's very nice work, of course, using deep networks to take the sort of video stream and turn it into features and ultimately prediction errors using temporal difference learning, just the sort that we've been modeling with the mice, right? So in particular, we used the deep Q networks actually used an A3C network to train the model to play the Atari game, this sort of standard famous DeepMind work, right? We just downloaded the code. But from our view, we can view the first n layers of this network, right, as basically the ones that have all the convolutional layers that are turning video input into value. Those end up with 512 neurons that represent something sort of highly processed about the task, and I just sort of don't care what it is. The last step of this really is a linear combination of these things to produce value and actions. And so it really is temporal difference learning, and if you sort of use the top n layers of this network, what you've got left is an example of a sort of highly varied high-dimensional phi, similar to, analogous to what we might find in the mouse brain actually playing the particular game that we're playing. So we can use this to sort of, exemplify how this all works, right? So in this task, so here's a sort of one trial of this game, right? The value function is plotted here. It's in units of sort of how many aliens I expect to shoot before I die. So over here, I'm expecting to shoot about 10 before I die, and every time I shoot one, it drops down a little bit. That's what you sort of see in this, and then it climbs back up when I still don't die. And then at some point, he gets unexpectedly shot, and his value expectation drops, right? So this is the sort of global view of value that this model predicts as it's playing one sort of episode of this game. The prediction error, for the same reason, overall looks like, hey, the game's starting, I'm going to shoot some aliens, nothing, 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 or little fluctuations. Oops, I got shot, and that's a negative prediction error. So that's the sort of classic, you know, it looks just like a Schultzian monkey, right? And indeed, who cares what's the difference? Okay, so these value functions in prediction errors, because they're based on 512 features phi and that bottom layer, we can use them to compute. These are the first 10 features. They do all kinds of things. Some of them care about the beginning of the task before the animal starts, or the animal, whatever it is, computer. Some of them care about other things. And those give rise to a bunch of prediction errors, which again do all kinds of weird things and respond at different times in the task, other than like when the task starts and when it ends, right? And this is 10 of them, and all I want to observe is that they look very different from one another, and together they add up to give you this, and these add up to give you this, right? So we can see how to decompose a sort of complex high-dimensional state into these sort of simple scalar, aggregate, decompose the simple scalar response into sort of complex high-dimensional features. And these prediction errors themselves, of course, by just the algebra that I showed you before, reproduce the sort of key effects from the Engelhard data. They have this sort of Q-like response, which is that they respond to their sort of particular feature phi I with a particular direction, of course, because of how they're computed. So they sort of track this tracks this and so on, or changes in this, the orange ones. And at the outcome time, because they all have the reward pumped into them, 85% of them, interesting number, covary with reward. So it sort of really does, again, sort of capture the salient aspects of the data. Okay, so coming back to the mice, or at least the mice in my computer, I want to meditate on this idea a little bit more. So let's notice some things about this. The first thing is, even though the individual prediction errors are an absolute mess and make you sad, the sum over all these prediction errors, if the model is right, should just be the sort of classic prediction error, the scalar prediction error, as illustrated here, and as you get from the classic models. And so, in some ways, this sort of actually is consistent with fiber photometry and voltammetry recordings that effectively aggregate over dopamine neurons and show nice prediction errors in animals. It's also consistent with the kind of early ePhys data where they tended to show average responses. Maybe the average dopamine response does look like a prediction error, even though the individual neurons are a mess, right? So if you do that in Alana's data, actually what you see is indeed, maybe I wasn't completely wrong sitting in my office, the neurons on aggregate really do respond more on easy trials than hard trials, sort of like the model predicts. The main difference actually is that there's this ramp, right? They also tend to ramp up, which the prediction error doesn't, along the track as you get more proximal to reward. And this is something that's been observed a bunch of times in other spatial tasks. And Sam Gershman has a paper about it, as about many things. And the point that he makes is perhaps, that really ties it, I think, into this work. I'm not sure you would say it quite this way, but one reason you might get a ramp like this from a classic prediction error signal is if there's limits in the function approximator, in the way the function approximator represents value across space, if that's constrained in some way that doesn't let it catch the right shape that the value actually follows, that's going to cause a prediction error in certain persistent prediction error even once you've learned the task, and in certain conditions that's going to ramp up. The question is why the state's constrained that way. What is it about the brain's representation of state or function approximation that causes this problem? And I actually think this work helps to shed insight into that in the following sense. So I think even more interesting than what the particular cues these dopamine neurons seem to respond to are, is how they sort of separate into different clusters. So these clusters aren't perfectly separate, right? There's some bleeding across them. But to a great extent, a population of dopamine neurons that know about speed, for instance, and don't know about position, and there's a population of dopamine neurons that know about position and don't know about speed, right? And so that implies a sort of factorization in the valuation, right, that is inappropriate, right? Because, for instance, in this task and in many tasks, the value consequences of where I am in terms of discounted future reward depend on how fast I'm getting moving, right? So I'm running to the end of the track. If I'm running fast, I'm sort of close to reward in time. And if I'm running slow, I'm farther from reward in time. And so the value function wraps up at different rates depending how fast I'm running. That means that value requires an interaction between position and speed. You can't possibly compute it, right, with neurons that know only about position and not speed, and vice versa. And I think that is, potentially you can sort of read this off Ben's data, that might explain the constraint on the state that Sam was, in a sense, looking for. And indeed, if you add that, if you factor the prediction error response in this model and you force it to not be able to represent the joint consequences of position and speed, you can reproduce this ramp. And I think that's the missing piece. Okay. The third point, and this is something we're sort of still working on, is these phi's, the model is sort of completely generic to what the features are. We haven't really said anything about that. We can sort of try to figure out what they are from the data in a sort of exploratory, sort of bottom-up way. But whatever it is, right, W is not arbitrary because W is learned by temporal difference learning. And so we should be able to predict, if we knew what phi was, or sort of guess what phi was, we should be able to predict both the kind of sign of W that goes with it, but sort of based on whether this is sort of a positive value thing or a negative value thing, and also sort of fluctuations from trial to trial in, if some phi is active on some trial and I get rewarded, then W should go up versus down. And so we should be able to actually see learning consequences of this representation, which we haven't quite managed to do yet. But I think that's interesting. One thing to say is it doesn't look, so whatever this W is, the model still sort of predicts that it should be the same for all the dopamine neurons that share the same phi I. So to the extent that Ben's clustering is the correct clustering, it does tend to be the case that, so for instance, all the neurons that care about position are positively modulated by position and not negatively, so that's sort of consistent. And most of the neurons that care about whether the previous trial is rewarded are positively and not negatively modulated, but there seem to be a few that go the other way. And so that is not consistent with this logic. We have to really sort of dive in and see if these few neurons actually represent something else, if their phi is really a different phi, or if something else is going on. But these are the kinds of things that we should be able to do with these data. Okay, so in the last few minutes, I want to try to connect this to sort of a broader line of work in these models. So the question arises, and I'm often asked this because this is, of course, mostly what I do is these sort of top-down kind of normative models. Why is the brain this way? And I think the first possibility is that it's just an accident. It's just a side effect, kind of a epiphenomenon or a serendipitous side effect of sort of non-uniform wiring around the circuit, and that it really wants it to be uniform. It just doesn't manage to sum it all up until it sort of gets back up to the top and striatum. That's still nice and interesting because it allows us to see from the dopamine neuron's perspective what they know about the input state in a way that I didn't think was going to be possible. So it's sort of encouraging, although it's not motivated, right? And of course, there are all kinds of reasons you might want to separate these channels, and particularly in a high-dimensional kind of function approximation setting, it can be very useful to be able to turn some of them off and manipulate them independently and stuff. And we can't do, obviously, full backprop in the brain, but if you have like different feature channels, you can and you're doing a task that involves your hands and not your feet, you turn off your feet and only work on prediction errors in your hands, and things like that. So that gives you a certain amount of, to the extent you have labeled lines, a little bit of control, which I think could be helpful in learning. Particularly in learning in a high-dimensional continuous situation where we can't ultimately rely on backprop to route errors around properly. Now there's actually a separate set of things that a bunch of people for many years, including me and Michael Frank, who was here before, have been saying, wouldn't it be great if we had multiple prediction errors because we really want to learn multiple value functions for reward versus punishment, or for different possible states of the world when we're uncertain, or for different levels of abstraction. There's a whole bunch of these ideas. I don't think these ideas are wrong, but I don't think they actually have anything to do with the results that I just showed you. So I think the results that I just showed you really do just kind of reflect this variation in the input, and they don't seem to require us to appeal to these more complicated kind of top-down schemes, or really to provide any particular support for them. I think there is support for some of these ideas at a sort of different spatial scale. So for instance, in particular, now Uchida really, I think, has the goods on a very separate population of dopamine neurons that are farther away that really care about punishments and not rewards involved in avoidance and so on. So I think that at a sort of larger separation, there really is some more organization going on that's not reflected in the kind of the local measurements we're doing here. But the local measurements we're doing here are really about something else. Okay, so that's it. What I've tried to unpack today is sort of one, I think, of the most revealing examples from literature of dopaminergic heterogeneity, which seems like it's really a problem for these models. And I think I try to tell you it's less than meets the eye and more than meets the eye. So it's less than meets the eye in the sense that it actually wasn't too hard just by realizing what ridiculous assumptions I was making sitting in my office with no mice anywhere, to reconcile these with the sort of scalar decision variables and sort of the beautiful theory behind common currencies and so on. And in particular, we really didn't need to appeal to, and I don't think we really found any support for, these kind of more structured multi-prediction error schemes where you have different prediction errors corresponding to macro actions and micro actions or something. Again, these schemes are not mutually exclusive, but I just think we're not zoomed out that far. There's also more than meets the eye, and it says that even though there's a sense in which the explanation for these findings is the same model that we've had since 1997, it shows us, I think, surprisingly, that this heterogeneity that is coming in from the population code for state, we're actually getting to see how the dopamine neurons see it sort of through a funhouse mirror. And that really gives us a window into a question that's been dogging these models for a long time. And I think will really help us kind of link it up going forward with the sorts of work that Mate was talking about this morning. And with the last question, which is sort of how does the brain figure out what features to use? And that's a thing, again, in different ways. Sam has worked on a lot. Tim, I think, talked about the other day and has worked on a lot, the DQNs. So I think we now are in a position to really work on the question of where do features come from and how does the brain represent and learn them, and how does that intersect with prediction error signaling. So that's what I have to say. Thanks a lot. This was work by Rachel, Alana, and Ben, funded by the Army. Thanks very much.
Thank you very much. So we have time for a couple of questions.
Hi, thank you. That was really interesting. I was wondering why you still interpret this as a, I guess the assumption here is that you're still adding up all these different components and then assuming that there is a final scalar prediction error signal towards the end. However, I guess through some experimental work, we know that these different signals project to different areas in the striatum. And is there any evidence that it actually sums towards the end, or rather they are just not summed up and just interpreted separately in these different populations?
Yeah, so I'm a little agnostic as to that. So it's clear that, and I'm involved in other work with Alana at the more area level, that it really is the case that the prediction error in the aggregate that you see in the dorsal striatum and the ventral striatum is different. And those differences are interesting. And again, those differences are sort of at a different scale than what I'm talking about. If you now zoom in on the ventral striatum, as it were, on the VTA, and then look at neuron by neuron, the aggregate signal of ventral striatum looks, actually we've done this with voltammetry also, looks a lot like in this task, what I showed you, the average over neurons, but the individual neurons still have a lot of variability in it. I don't know to what extent the neurons in striatum, the recipient neurons, can see that variation, or if it gets all kind of averaged out by population, by volumetric signaling and stuff. It could go either way, and to the extent that they remain separate, and that's interesting and has consequences, they're not sort of fatal for the model. It's not like I'm relying on that. I'm sort of saying the very most baseline model from 1997 actually maps pretty well onto this. If you want to imagine that it somewhat separates out as well, even in the local scale, that's got measurable consequences we should be able to find out, but I'm not. It could go either way.
Hey, thanks for the really interesting talk. In the model you proposed, you kind of had just a sum over all the kind of individually computed values for each of the individual features, right? So this is kind of averaging across all the features. And near the end of the talk, you kind of hinted at the fact that maybe some other models, such as like something like the successor representation, is kind of learning the right features to use. And do you think that this kind of feature engineering that's been happening in other parts of the brain essentially allows that each of these individual feature values for each of the features to essentially be equally weighted in the model that you have, right? Because essentially you just have this averaging across all the features. So is the engineering there also allowing us, teaching the animal which features are valuable, essentially, and essentially equally weighting them across the different aspects of the state vector?
Well, I think there's two things. One is that, of course, without looking at the rewards, you don't know which ones are relevant for value. That's part of the problem. It's easy, again, for me to sit around and say all that matters for this task is the running count of towers, but the mouse doesn't know that, certainly not on day one. And so presumably, the brain has got lots of features in it, and part of dopamine's job is to try to correlate those against value and figure out what weights to put on them, those W's. So it's not the case that they're not, in a sense, averaged uniformly. The job of dopamine is to figure out which are the ones to pay attention to and which not to. That's part of the answer. But the other part of the answer is I do think there may be insights that we can gain if this is really the architecture, if you sort of bottom out in a linear stage where you've got to sum up a bunch of features starting from a more complicated perceptual analysis, there may be things you can do to make sure that the sum, that you don't do things like the mistake that I think the neurons are making of separating position from value when they really position from speed when they really interact. That poses constraints on the representation that's desirable in cortex as well, right? So figuring out, and I think that interacts with the success representation as well in ways that I would hand wave to unpack right now, but I'm happy to talk about. So I think thinking about this will help us think about how the brain should learn the features as well in a way that would help make this work.
One more? Oh yeah, so in the middle.
Thanks, really cool stuff. So given that your final results seem to be still going along with the kind of scalar output idea, is it fair to say that that means it doesn't really make any new predictions in terms of more tonic signals, and it doesn't make any new predictions in terms of pharmacological manipulations?
I mean, I think pharmacology is sort of always based on the scalar model, right? Because it's so crude. So I don't think we'd have anything more to say about that. I think in terms of tonic activity, I think kind of zeroing in on the interaction between the state representation and this ramping stuff actually is a sort of new potential for understanding some of this stuff. So I think, again, I think a great deal of what's emerging from Josh Burke and from Anne Graybiel and others is that at least in these spatial tasks, there's all this sustained activity, this sort of ramping activity as animals run around these mazes, and you can see that in our data as well. And that's a real puzzle for the prediction error models on one level, and I think Sam is right that really intersects with this question of function approximation. So as we understand how that works, I think it's going to help us understand these ramping responses, and so I think that is a new aspect of it.
Okay, thank you. Thanks a lot.
Thanks.